Thank you very much. So yes, I'm Esther, and I will be reviewing the last strategies on reducing inflammation and fibrosis in Duchenne. So I'm part of the John Walton Muscular Dystrophy Research Center. We are based in Newcastle, and it's a multidisciplinary center that we work all together to try to find a cure for Duchenne and other dystrophies. So just a brief introduction about what is fibrosis and what is inflammation. These both two phenotypes appear in the muscle during the progression of the disease. So what we know so far is that in Duchenne boys, the absence of dystrophin in the muscle promotes a continuous damage into the tissue. So that chronic damage into the muscle promotes the increase of necrosis and degeneration of the muscle, and this leads to a different phenotypes into the muscle. So there are different changes that come from that absence of dystrophin. One of the first changes that we can observe is the increase of the inflammation. So there is an increase of different inflammatory cells into the muscle that try to repair that continuous damage. Inflammation, it's a response to that damage, but this increase of inflammation is followed by an increase in fibrosis and adipogenesis in the muscle. So if we compare these two slides, we can see that in the healthy muscle, the architecture of the muscle is more or less the same. This represents the muscle fibers, and in the Duchenne one, we can see that there is more white area that represents the increase of adipogenesis. There is also a different size of the fiber tissue. So we know that that increase of adipogenesis and fibrosis in the muscle impairs the proper function of the muscle, and it's something that will have some consequences into the patient. So if we look more in detail, in early stages of the disease, the muscle in the patient is more or less preserved because that damage of the absence of dystrophin has not been really done. But as the disease progresses, there is an increase of the inflammatory response in the muscle that is characterized by these small dots that you can see in the image. These represent different inflammatory cells that migrate into the muscle. And this is led by a replacement of the muscle of fatty and fibrous tissue. So as the disease progresses, there is more fibrotic and adipogenesis tissue in the muscle. Although we know that the muscle degeneration that occurs in the muscle is due to the absence of dystrophin, we don't know exactly what makes that progression to keep going into the muscle. So we know so far that in the muscle, apart from the muscle cells that will make the muscle fibers, there are other type of cells that are involved in that regeneration of the muscle. So for example, there are pericytes that are cells that are around the blood vessels. We have satellite cells, which are the precursor cells of the muscle fiber. And we have also fibroadipogenic progenitor cells that we can also call them FAP, that we know are the cells that will develop the adipogenesis and fibrosis. So there is a lot of investigation going now, trying to identify which are the mechanisms that regulate those cells in Duchenne, because although we need all these cells in healthy muscle to promote the homeostasis and the proper regeneration of the muscle, we know so far that in Duchenne there is an increase and an overactivation of these surrounding cells that will promote that increase of adipogenesis and fibrosis and inflammation into the patients. So if we go to the current therapeutic strategies, we we can see that those can be divided in two different lines. The first line are dystrophin therapeutic strategies that aim to restore the expression and function of the dystrophin. So these types of therapies include gene-based therapies, cell-based therapies, and protein replacement therapies. And the next talk will be focused on that type of therapies. But the second line is the therapeutic strategies that aims to improve the muscle function and the quality of the patient by targeting the pathological changes that we were talking before. And although it seems that there are two completely different strategies, the combination of both seems as the most promising therapy. And why that? So with gene therapy, we know that we are going to restore the gene in the muscle, dystrophin. However, what we know is that the delivery of that type of therapies, it's not as easy to be targeted in early stages than later stages of the disease. So in early stages, when the muscle is still well preserved and there is not a lot of inflammation, fibrosis, or adipogenesis, the delivery of the gene therapy is more efficiency because it can target the muscle. There is more muscle tissue, so it's easier to target the muscle. However, as the disease progresses, we lost a lot of muscle tissue and we have more fibrogenesis, adipogenesis. So it's more difficult for those type of therapies to arrive to the muscle because there is less content of tissue. So in front of that situation, we believe that antifibrotic and anti-inflammatory therapies are a good candidate for this type of treatments. And we think that therapies that can slow down the progression of the disease can be a good combination for gene therapy to maintain the early stages of the muscle and then include the gene therapy. So moving into what are the current therapies going on in inflammation and fibrosis, just to briefly mention that the different phases that we do in the lab and then in the clinical trials to approve new treatments. So we first of all start in the preclinical stage, and so before any treatment is tested, in the lab what we do is just try to see the effect of the drugs and the treatment in vitro phases. So we use a lot of human cells just to see the effect of that new treatment, and we also use in vivo models. So normally for Duchenne, we use the MDX mice, that is the mouse model that represents the Duchenne phenotype. And the characteristic of those mice is that, as same as in patients, it doesn't have the dystrophin gene. But although a lot of clinical trials and preclinical research is doing into that mice, the phenotype, it's not as good as the patient. So mice have a better regeneration of the muscle in general, so it's not the perfect model to try these new treatments. For that reason, there are other type of models that are being investigated now, like for example, muscle on a chip, which is like a 3D cell culture model in which we can create more or less a 3D muscle from patients, from cells from patients, and see how this muscle on a chip reacts to the treatment. So once we start in the lab and trying these new treatments and find good candidates, the next phase is moving into phase 1, where the safety and the side effects of the new treatment is tested, and this includes a small cohort of patients. In phase 2, the safety and side effects of the dose are also studied, and here we can find that the cohort of the patients is bigger, and if the drug has tested safety and in the patient, it goes to phase 3, where it can be compared to the current standard treatment. And the drug keeps being investigated for their safety and side effects. If finally that drug is approved, in phase 4, it's just to continue investigating the drugs, because we will know the long-term effects of that drug. So it's something that also gives us a lot of value. So regarding the therapeutic strategies, I will be focused on the inflammatory and fibrosis therapies, and I will just briefly talk about some of these drugs that has been investigated in the last years. So what is inflammation? Inflammation is described as the process by which our immune system recognizes and removes harmful and foreign stimuli and begins the healing process. And in normal situations, inflammation is an acute response to something that is foreign to the body. However, in Duchenne, we know that this is a chronic state. And what you can see in that graph is just different cells trying to recognize the harmful and foreign stimuli and try to attack them. So what occurs in Duchenne is that the continuous contraction induced damage that are in the muscle induce different responses. So one of them is that the membrane is more susceptible to release different factors from the muscle, and this is something that will recruit the immune system. So as you can see, the muscle that is represented on the top of the image has like the dystrophin, the absence of dystrophin makes that membrane more leaky. So there will be a lot of release of different factors that the macrophages, which are a type of immune cells or neutrophils, will recognize and will create that inflammatory response. So some of these factors released by the muscle can be called like TNF-alpha, interferon gamma, and there are a lot of therapeutic strategies that we will see now that target this type of molecules. So currently, the anti-inflammatory treatment for Duchenne is the use of glucocorticoids, but although they have proved that it can slow down the progression of the disease and improve the pulmonary function and delay the onset of cardiomyopathy in patients, there are also several side effects for long-term treatment. And so for that reason, there is ongoing investigation trying to find another type of anti-inflammatory drug that can reduce these side effects. So just as an example, in phase 1, some years ago, there was flavoxidine, which was an anti-inflammatory small molecule that what does this drug is that it can recognize some of that molecules that are leaked by the membrane and can inhibit them. So although in phase 1 indicated that the treatment was well tolerated, the outcome measures failed to show significant effects for short-term treatment. In phase 2, we also had Increlex, which is a recombinant human insulin growth factor. And that drug improved the muscle function and the regeneration of the muscle when it was tested in the mouse model that represents Duchenne. However, the therapy had no effect on muscle function in patients. Another one in the same line is TAS-205 that inhibits prostaglandin D2. And that inhibition showed like that it was beneficial in the mice model. However, in the phase 2, although it was safety and well tolerated by the patients, the effectiveness of that drug was not completely approved. In phase 3, we had different drugs. So this one is a salanoxin. It's a small molecule that inhibits NF-kappa beta, another inflammatory molecule released by the muscle. And in phase 3, this drug found to be safe and tolerable by the patients, but the improvement of this patient was not completely clear. Tamoxifen was another one that it's a selective estrogen receptor modulator that improved the muscle 4 in the MDX mice. But in phase 3, although it was safety and well tolerated by the patients, it failed to delay the disease progression of the patients. And finally, givinostat, it's a histone deacetylase inhibitor that it's currently being developed for the treatment of Duchenne. It's still on clinical trials, and in phase 2 showed that it could increase the number of muscle fibers and suppress fibrosis. And in phase 3, the evaluation of givinostat also that it can also slow down the progression of the disease in Duchenne patients. So this is one of the drugs that it's still ongoing in clinical trials. And vamorolone is another type of glucocorticoid that it's similar to the current treatment. However, this has reduced side effects compared to prednisolone. So this reached the phase 3 in clinical trials, and I think it's not been accepted yet, but it's two of the drugs that are still in phase 3. And the success of that drug in Duchenne patients and also prompt to an ongoing phase 2 pilot trial for Becker muscle dystrophy. Okay, so now moving into the fibrotic therapeutic strategies. Fibrosis, as I was saying before, it's kind of a consequence of the active inflammatory state in the muscle. So fibrosis is described as a pathological change in DMD. It's a direct response of the chronic tissue injury, and it's an uncontrolled wound healing process characterized by the excessive accumulation of extracellular matrix proteins. We can see the extracellular matrix proteins in the pale pink area of that slide that represents the increase of fibrosis. And the excessive accumulation of that matrix in the muscle impairs the functionality and the structure of the tissue. And as the disease progresses, we can see that that pale pink area increases over time. So there is an increase and accumulation of fibrosis over time. So some examples of some treatment that has been developed for using as antifibrotic in Duchenne. One of them is losartan. This is a drug that was used to treat high blood pressure, but also when mice models were treated with losartan, and it showed that it decreased the fibrotic area in the diaphragm, and it restored the muscle strength. However, the clinical trial in patients in phase 1, although it significantly improved the cardiac function in Duchenne, it didn't show an improvement in reducing fibrosis in the skeletal muscle. The second one, allofuginone, is another small molecule that decreases the established fibrosis in the MDX mice. However, the safety and tolerability was not approved in the phase 1 clinical trial. Pamrevlumab, it's a phase 3 drug that was produced by Fibrogen, and it demonstrated safety and tolerability, but in phase 3 assays, it didn't show a proper delay of the onset of the disease. And finally, as some preclinical drugs that has been investigated, we have soramine, which is a TGF-beta inhibitor. TGF-beta is one of that molecules that increases fibrosis, one of the master molecules, I would say, that increase fibrosis in the muscle. Imatinib is an anti-neoplastic drug that also showed the ability to inhibit some pro-fibrogenic factors. And Remicade, which is a treatment that it also inhibits TGF-beta and TNF-alpha, another target molecule of fibrosis, but none of them show a positive response in mice models. So as a summary, these are some of the drugs that has been investigated. And I would like to highlight the preclinical ones because although we have seen that a lot of them didn't arrive to being approved or to phase 3 or 4, the molecular mechanisms of those drugs are really important in terms of knowledge because that help us to know which mechanisms are differentially regulated in the muscle and how we can target them. And even that some of them didn't arrive to other phases, something that can be done is to create analogs of those drugs, so small modification that can improve the efficacy in the muscle and also to improve the efficiency of these drugs into the muscle. So also what we are investigating now in our group is some other molecules or some other mechanisms involved in the increase of fibrosis in order to try to decrease that phenotype in the muscle. So as I was saying at the beginning, I work in the John Walton Muscle Dystrophy Research Center. More specifically, we work in the basic research strand. Our group is led by Professor Jordi Diaz-Manera, and what we always try to be focused in our lab is to understand the progression of muscle dystrophies. So we know so far that early stages of the disease, the muscle is well preserved, and in later stages there are a lot of different changes that can impair the proper architecture of the muscle. So one of our principal aims is trying to understand which are those differences in the mechanism of and the progression of the disease, because that will give us some clues to which type of drugs are going to be more effective in the muscle. So, for example, here in the first, on the top of the image, there is a representation of the muscle slides of a healthy, healthy sample. So this muscle has had an acute injury that can be doing a sport or doing another type of activity that creates an acute injury into the muscle. So in that example, we can see that there is an increase of the fibrotic and the inflammation, but that increase after small time, it's reduced. So it's a transitory phase that we all need to regenerate the muscle. However, this process, it's not reduced in Duchenne. In Duchenne, what we can see is that there is a progressive extracellular matrix deposition, so an increase of the fibrotic tissue into the muscle that will impair the proper architecture and the proper function of the muscle. So we are trying to identify which mechanisms are dysregulated in this type of changes. So we normally we are in the preclinical phase, I would say, and we work with muscle tissue from patients. So we are very lucky because we work close to the Royal Victoria Hospital, so we can get muscle biopsy from patients and also from healthy individuals. And what we do is that we collect that piece of muscle from the hospital, and we can put that in cell culture and then obtain lots of different cells that reside into the muscle. So we can sort this muscle explant. So with that cell sorting, what means is that we can select the different type of cells that are in the muscle, and we can have a batch of different myoblasts, for example, which are the precursors of the muscle tissue. We can have FABs, which are the precursors of the fibroblasts and adipogenesis. And with that sample, we can do different tests. This has allowed us to recognize different mechanisms that are involved in the increase of fibrosis in the muscle, and also to try to find some molecules that can reduce that increase of fibrosis. We also work with the biobank from the John Walton Muscle Dystrophy Research Center. In the biobank, we have samples from over more than 123 different rare diseases, and this is great value for us because we can work directly with human tissue, which is something not easy, and of course it makes the translation of the research more accurate. So one of the examples that we can do with this muscle biopsy is just we can cut the biopsies into small pieces. We put those into a petri dish in cell culture, so in proper conditions for the muscle. And from that, we obtain lots of cells that we can keep expand them. And after the selection of the ones we want, in this particular example, we got the fibroadipogenic progenitor cells, so the FAB cells. And with FAB cells, we have a protocol and that allows us to differentiate those cells into adipocytes or fibroblasts. And this is important because adipocytes are the cells that will lead to the adipogenesis tissue in the muscle, and fibroblasts are the cells that will lead to the fibrotic tissue in the muscle. So we have the precursor cell that will increase this type of different tissues. So that differentiation of FABs into adipocytes or fibroblasts has allowed us to start different projects trying to find a drug or a treatment to slow down the progression of the disease, so to inhibit that increase of adipogenesis or fibrogenesis. So just as an example of an ongoing project that we are doing now is in collaboration with Duchenne UK, and we try to find potential drugs that can stop the progression of the disease. So in the first phase of this specific project, we tested more than 500 compounds. So we have large libraries of already approved drugs that have been approved for another type of diseases. So we think that it's better to have this type of drugs, as then it will enter easily into clinical trials because they have been already approved for safety and tolerability. So with that large library, what we did is that we culture the cells that we obtain from patients in this type of plates, and then we set up a high throughput screening. So that means that we could test all these compounds into this type of plates. So every dot that you can see in this image will represent a different drug. And with that mechanism, we could test how many of these drugs were able to reduce the differentiation of Fabs into fibroblasts or adipocytes. So how many drugs could be used in the future to target the progression of the disease. So in the first stage, we started with 500 compounds, and from that high-throughput screening, we narrowed that big library into 8 final drugs. So with these 8 final drugs that we observed that could have effect in that differentiation process that we were talking about, we did more in vitro studies. So we did toxicity studies into cells just to see how the cell react to this treatment. We also analyzed the cell effect of the treatment of that drug, and we also optimized which dose should we use to see a positive effect in the cell without affecting the viability. In the next step, we are now in the preclinical studies. So we are using, from that large library, we are finally using 2 drugs that we have seen a potential effect. So we are using a mouse model that represents Duchenne, but in this case, it's not the classical MDX mouse model. It's another one that represents more the fibrotic phenotype. It's called D2MDX. And what we are planning to do is to treat these animals with these drugs, and over time that they are being treated, we can do some muscle function tests into the animals, like grip strength, we can do treadmill, and we can do the hanging wire, and we can analyze if the force of those animals is increased with the treatment of the drug. So if that drug has an effect on the response of the animal. And by the end of the treatment, we can also analyze the histology of the muscle. So we can do slides of the muscle as the one that I was showing before to see if that fibrotic area is reduced, if the adipogenesis area is reduced, and also if the inflammatory area is reduced, and how the fibers are, because if we reduce the fibrotic and adipogenic area, we are promoting the regeneration of the muscles, so fibers should be more similar than to a healthy muscle. So we believe that if positive results leads up from here, potential clinical trials in the future could be tested in patients, and we believe that finding some drugs that can stop the progression of the disease can be a good combined therapy for the genetic therapies. So finally, I would like you to introduce another type of project that is ongoing now in the John Walton Muscular Dystrophy Research Center. This has nothing to do with inflammation and fibrosis, but it's for the diagnosis of the neuromuscular diseases. The project is called MyoGuide, and it's meant to be to apply artificial intelligence to the analysis of muscle MRI in neuromuscular diseases. So just an overview of the project. It's a supervised machine learning that has proven to be helpful in the analysis of muscle MRI to guide the diagnosis. And to make it easier to understand, basically what we are doing with this machine learning is just training a software to diagnose different patients. And the main objective of that project is to improve the diagnostic algorithm by increasing the number of MRIs and muscle disorders that can be predicted in the algorithm. So basically, what is that from all the MRIs from the different neuromuscular disorders, the software can recognize the pattern of this MRI and can identify which is more similar to each type of disease. So the aim of the MyoGuide project is to be free of use for clinicians around the world, And basically, it would be to upload a scan MRI, and the software, the artificial intelligence, will present the most likely diagnosis that then of course will be confirmed by genetic tests. So basically, we plan to reduce the time of diagnosis by the neurologist by using an artificial intelligence. So for the development of this type of project, and the researchers, what they did is they upload different scans from different patients that there were already stored in the NHS, and then the machine can identify some specific patterns for each type of disease. So MyoGuide has all the regulatory approvals and confidential advisory group, and this means that we can use the patient data that has been already consented before. So if you as a patient or as a familiar of a patient have chosen not to share your data, then these scans and this data is not used. And the data that the software is using, it's completely anonymized, and it has just the score of the MRI scan, the age, sex, and the genetic diagnosis. For example, male, 10 years old, Duchenne. This is an overview of the current collaborators of the project. And in light blue, we have the potent collaboration. And the aim of this project is to have, of course, more collaborators that can upload all the scans and improve the machine learning. So finally, I will really appreciate if you can give feedback about how do you feel about sharing using the data that you have already consent as a patient or as a familiar of a patient for keeping the research on this type of projects or on other type of projects, because this is really, it's really helpful for us to keep ongoing on the research and to keep trying to find treatments or also shorten the time of the diagnosis. So in the first QR, there is a longer video by Professor Jordi Diaz-Manera that explains more in detail that project. And in the viewer one, there is the survey that is just a 1-minute survey. I have some cards that I will leave after that in the registration desk, so you can scan the code. And that's it. So thank you very much for your attention.